A warm welcome, dear children. In the previous video, we had talked about mitosis in plant cells. So, we studied the various stages of mitosis in plant cells, and now the second practical that you are supposed to do is to study mitosis, the various stages of mitosis in animal cells, and of course from the permanent slides. And grasshopper is the specimen that is taken. We will study the stages of mitosis in grasshopper. So, now coming to the requirements for this particular practical work, spotting, we require a microscope to view the slides, right? So, these are the slides here. Then we require a practical notebook where you can copy down your work, right? Then you need a laboratory manual. Other than this, you require a ruler, you require a pen, a pencil, eraser and a sharpener, right? So, you will be drawing diagrams, etc. So, those are the requirements for the practical. And to observe, right, the procedure is that you will take each slide and you will put it on the stage of the microscope. Here you can see this is the stage. Here these are the clips. You will place the slide here under the microscope, right? These are the knobs, adjustments with which you will adjust to the lower power of the microscope, and then you will observe the slide under the high power of the microscope, right? So, you will ensure that the slide is placed properly, and this is the eyepiece from where you would be able to view the slide under the microscope, right? So, of course, when you will view under the low power of the microscope, then you will be able to just see the cells along with the nucleus. Otherwise, when you observe it under the high power of the microscope, you will observe the following stages, and of course, the different stages of the mitosis would be given to you. So, we will have a quick review of mitosis in animal cells. Coming to the first stage, that is, we can say a phase in between two mitotic divisions, is the interphase, and the cells contain a prominent nucleus, right? So, if we look at this structure here, so there's a prominent nucleus with intact nuclear membrane. So, if you look at this, you can see the nuclear membrane here is intact, right? And the nucleolus also being present, right? Chromatin material appears in the form of a network. So, this is just a diagrammatic representation, right? Next stage is the prophase stage, and the chromatin material shortens, condenses into thread-like structures called the chromosomes. And the chromosomes consist of two chromatids. They are joined by centromeres, right? So, here this region here is the centromere that has been labeled, right? So, that is joined by the centromere. The centriole pair replicates to form two pairs of centrioles. So, you can see here these are the centriole pairs. These we are talking about, they are present here. So, they then start migrating to the opposite poles, right? So, they will start moving. You can see here in the first figure, they are more closer, and in the second figure, that is the prophase stage, you can see they are moving apart. So, they are moving towards the two poles, and then you will see that the nuclear membrane and the nucleolus, they start disappearing. Next stage is the metaphase stage, where you can see a bipolar spindle develops. Now, if you look at this structure here, these are the spindle fibers, right? So, these are the spindle fibers. Bipolar at the two ends, these are the polar ends, right? So the bipolar spindle develops in the cells with asters, right? So asters, we'll see it in the next figure, the next diagram that I'll show you of the asters, right, that are formed. Chromosomes are arranged at the equator. You can see here at the equatorial plate how the chromosomes are arranged, and they are attached to the spindle fibers with the help of the centromeres, right? So the next stage is the anaphase stage, right? The sister chromatids of each chromosome, you can see how they separate from each other and appear in either V-shaped, mostly they are here shown as V-shaped, so either we can see it V-shaped, L-shaped, right, or J-shaped, depending upon where the centromere is present. So they start moving, you can see here, they are moving towards the opposite poles. And then comes the last stage, which is the telophase stage. And in the telophase stage, you can see that the daughter chromosomes, they uncoil to form chromatin fibers at the two poles, and the nuclear membrane, you can see here, it starts reappearing. In this figure, you can see it starts reappearing, right? The constriction would be here, which you would view in the next diagram. It starts deepening, right? And the parent cell divides into two. So that's about the mitosis, which you can see here very clearly here in mitosis in animal cells. You can see the first stage is the interphase stage, and you can see the plasma membrane. You can see the centriole here, which splits and moves into the two opposite poles, right? The centrosome here, centrosome, and inside are the centrioles. The two dots are the centrioles, right? You can see the nucleolus, very tiny nucleolus shown over here, and all the nuclear membrane is intact, and inside that you can see the chromatin material all arranged in the form of a network like this, right? Then the prophase stage, you see, prophase is marked by the disintegration of the nuclear membrane, right, loss of the nucleolus. You can see how the chromosomes, they become condensed from thread fiber like this to becoming a little visible like this. There's a difference in the shape, you see, right? So they become condensed, and then you can see the formation of aster, right? See, what happens is that the two centrioles which were found over here like this, they move towards the opposite poles, right? The centrioles, the pairs, they move towards the opposite poles, and aster formation like this starts taking place. From here you can see how the spindle fibers are formed, right? So formation of spindle fibers take place. Then if you see the next stage, which is the metaphase stage, at the metaphase stage you can see how the chromosomes, they arrange themselves on the equatorial plate, right? And the last stage, if you see here, is how this is the telophase stage, right? You can see the chromosomes, they have split into two daughter chromatids. Half is moving towards one pole, the other half is moving towards the other pole, right? And then you can see once they have moved to the opposite poles here, the nuclear membrane forms again. You can see that dot is the nucleolus that is formed, and you can see how the cytoplasm, it starts deepening. You see, cytokinesis has to take place here. So cytokinesis is division of the cytoplasm. So a furrow like this is being formed, right? This furrow will go on deepening. It will go on deepening till it divides into two daughter cells, right? So that's about mitosis in animals. Here again you can see very clearly all the stages of mitosis in animals, right, in the animal cells. These are the slides which you can have a view of the various stages in the form of a slide over here. So we come to the first stage. This is the interphase stage, right, where the chromatin material is all coiled up. Next you can see here is the prophase stage, where you can see the chromosomes, they start thickening over here, right? Disappearance of the nuclear membrane and the nucleolus will take place, right? Next is the metaphase stage. Here you can see very if you'll have to strain your eyes a little to see the centrioles to the opposite poles. You can see very fine in white color, these are the spindle fibers, right, that are formed. Aster formation. This is the equatorial region where the chromosomes have arranged themselves in the metaphase stage. So the metaphase stage is actually characterized by the arrangement of the chromosomes on the equatorial plate. Anaphase. Anaphase is the stage where the spindle fibers, they start contracting, the chromatids, they split and they move towards the opposite poles, right? And then comes the next stage, that is the telophase stage. They have moved towards the opposite poles and the cell furrow starts deepening, starts here. This will go on deepening till this leads to the formation of two daughter cells like this, and which will again undergo the interphase stage. So that's about mitosis in animals. This is the various stages again in different forms. I've told you, I have tried to show you where you can locate the various stages, very nicely shown over here. This is the interphase stage, right? You can see the thickening starts taking place. Prophase, metaphase, spindle fibers are formed, centrioles are there, asters are there, right? Nuclear membrane has disappeared, nucleolus is not there, and the chromosomes have arranged on the metaphase. Anaphase stage, you can see the splitting of the chromatids, sister chromatids, and movement towards the opposite poles by the contraction of the spindle fibers. Telophase, the chromosomes have moved towards the opposite poles and the cell furrow starts deepening. Cytokinesis takes place where two daughter cells are formed. Now a very important part is what is the difference between mitosis in animal cells and mitosis in plant cells. So if we look at the major differences, mitosis in animal cells starts taking place by the deepening or the formation of a furrow, right? You can see here this furrow is formed, which is a depression which goes on increasing, right? So cytokinesis in animal cells involve furrow formation between the daughter nuclei. So this way it divides into two daughter cells. Whereas in plant cells, furrow cannot be formed because of the presence of hard cell wall. So here this is hard, this part is very hard. So this will not deepen. So furrow will not be formed. So if furrow is not formed, then what happens? If furrow is not formed, then there is a cell plate formation that starts taking place, right, in plant cells. So cell division involves the formation of cell plate in plant cells and furrow deepens in animal cells. Now what is this cell plate made up of? It is formed by phragmosomes and phragmoplast, whereas these are not present and there is no cell formation that takes cell plate formation that takes place in animal cells. Now what are phragmosomes or and phragmoplast? You can see here phragmosome is nothing but it is just a kind of it is just the cytoplasm that is there cytoplasm that is there, and more formation of the cytoplasm that takes place. That is just what is the phragmosome, and this goes on increasing. There is a band of phragmosome that is formed in the cell, right? So that deepens, that is what forms the cell plate. Another difference between animal cell and plant cell mitosis is that centrioles are found inside the centrosome. So what we saw at one pole was a centrosome inside the centrioles, whereas these centrioles and centrosomes are absent in plant cells. They are present, centrioles are cell organelles present in animal cells which are absent in plant cells. So in animal cells, spindle fibers are formed by centrioles. Now you see these centrioles, they move towards the opposite poles, and spindle fibers are formed from these centrioles only, right? Since there are no centrioles in the plant cells, so spindle fibers assemble without the centrosome. So here you see there are no centrosome or centrioles that are present. Next is the formation of aster. You will see that aster formation, star-like, that takes place only in the animal cells, whereas this aster formation is not found in the plant cells. So spindle fibers in animal cells formed by centrioles show aster arrangement. What is this aster arrangement? They look like a star giving out rays. So that's a centriole and something like this, right? That is the aster formation. So that's about all the phases of mitosis in animal cells. And of course you must remember that the two cells that are formed are diploid cells in mitosis. Precautions. Precautions are again the same. You use fine adjustment knobs while focusing in high power of the microscope, and the objective lens should not touch the slide. So that's all for this class, children. Thank you.